I'd like to talk to you about how we can develop computational simulations for tissue engineering. And I think the key message really for me in this time is to see how we can use computer simulations that is complementary to experimental methods, and how computational simulations can actually inform to do better experiments. So just for the ones who are not very familiar with tissue engineering, this is the basic concept here, that if you want to regenerate a new type of tissue, you'd want to extract some cells. So for example, here this is the case of a large gap defect in the bone, and you want to isolate the cells, expand them into a bioreactor, and seed those cells into a scaffold, which is basically a porous material, which is represented here. Now, those cells need to be well distributed within the scaffold so that they will produce extracellular matrix, and then you can put it back into a patient with the scaffold. Now, this seeding part is very important because this is what's going to induce all the rest of the process in terms of cell differentiation and cell matrix formation. So, and it's largely influenced by mechanical stimuli. So normally you would put that scaffold into a bioreactor and stimulate the cells under fluid flow conditions or mechanical loading. And really what we want to be able is to see whether we can actually improve that seeding process experimentally, so in vitro. So the research questions that I wanted to address was whether we can actually model this perfusion seeding process so that we could improve then the cell seeding efficiency in vitro. So what I've done is that I've selected a material which was produced using additive manufacturing techniques. So you've got a very regular scaffold. This was made by 3D Biotech. In principle, it's very regular. Now, it turns out it's not as regular as this. Might be difficult to see in the table here, but this is the number of samples here, which you can see different parameters that have been characterized using micro-CT and ACM. And there is some variability within the sample. So for example, in particular, if you're looking at the fiber diameter, some samples are very low fiber diameter, 254 microns, while others are close to 400. So, and this is just an average, so you're going to get quite large variability. And you can see from the picture here that there is some variability that you can already see from the naked eye here. Now, this is quite a crude characterization in some way, and we thought that maybe we can use computer simulations to actually better characterize that in a more quantified manner. So what we've done is we've taken the ideal scaffold, which is represented here with those dimensions, and what we wanted to do is to be able to compare then the ideal scaffold with actually what has been produced, so to verify whether the quality of production was right, and also to see whether there was some repeatability if you're taking different types of scaffolds. And I'm not going into detail because it's already been published here, but I just want to show you some of the results. This is basically what you see here is the velocity of fluid flow. So we simulate fluid flow passing through the scaffold, and this is showing at the top of the scaffold, and then we're going down at the bottom of the scaffold here. So this is for the ideal scaffold, and you can see the fluid flow is very homogenized, which is kind of what you would expect from the CAN model. Now this is for real scaffold, and you can see that there is some variation that happens here, but it's quite good. If you compare with 5 different scaffolds, you can see quite, the S3 has very high fluid flow, so 10 times higher than what you actually would expect. So the cells, that means the cells will sense a fluid flow that is 10 times higher than what is the input that you wanted to give to them. If you move down to the scaffold, you'll see that large variability. And one scaffold that was good, at the top here is getting actually very bad in the bottom. So that's going to affect the viability of your experimental results as well. Now, once we've quantified basically the morphology of our scaffolds, we wanted to see whether we could simulate then the cell seeding process. So we have developed some computer simulations where we simulate the chamber. So normally the scaffold is held here within a tube, and this is the chamber basically, and we have a perfusion system that brings the fluid flow from here, that goes passing through the scaffold and then goes there, and we reverse the fluid flow so that the cells can also attach within the scaffold back. And it goes back and forth, so we've got many cycles happen. So we can simulate the cells. So those are, what you see here in blue is the cells that are actually going through the scaffold. And whenever they basically touch the wall of the scaffolds, then they attach. So we can actually simulate where the cells are going to attach here. We can vary the type of fluid flow conditions. So, we can go from a very low fluid flow, 12 microliter per minute, to a very high one. And what's interesting is if you have very low fluid flow, what you see is this is the fluid flow patterns here in blue. So it passes through the scaffolds, but the cells are actually falling off here. So there's not enough convection for the cells to pass through the scaffold. If you've got very high fluid flow, like 600, it happens that you've got some recirculation areas here. So it's not very good either. So there is some kind of an optimum fluid flow rate for this scaffold here. Now, if you do all the analysis and you compare with some of the experiments, so you've got simulations in blue here and experiments in green, then you can see that the trend is relatively good. So between those two here, but the general message is that the cell seeding efficiency is very low, actually, whatever is the fluid flow rate here. If you compare to static, which is basically just seeding cells like this with a syringe, it's much lower. So it's not working very well, really. We're not getting very much cells inside. So what we did is to actually change the bioreactor configuration by having the scaffolds that is rotating. So we've got fluid flow that is coming in from the top, going down, and at the same time we've got a scaffold that is rotating around here. And what we expected is that would actually improve the chances of cells to actually reach contact with the scaffold. So we've looked at many different types of rotational speed, from no rotation at all to 100 run per minute, and this is the fluid flow profile you can see here. And you can see that starting from around 10, you're starting to see quite different effects here, where you've got a lot more homogeneous fluid flow here. Now, some very high fluid flow, you've got some recirculation areas happening again here. Now, what's interesting is to see how this affects the cell attachment. So what we've been looking at is, in purple here, is represented the cells that are within the scaffold. And as you can see, with low fluid flow, there's not many cells actually being attached here. Once you start around 10 1 per minute, then you've got actually fairly nice cell attachment with very nice distribution. If you increase the fluid flow, there's more cells actually being attached, but then you start losing a little bit in terms of distribution. So there seems to be some kind of optimum flow rate between 10 and 60, which would need to be better defined. Now, the good thing is that you can actually predict that the cell seeding efficiency would be around 99%. So we've increased now cell seeding efficiency that was below 20% to close to 100%. So that shows that, by actually designing then a new bioreactor, then you can actually improve very significantly the cell attachment, and that can inform then the in vitro experiments. So just going to my conclusion, what I wanted to show you is that first is that scaffold fabrication. I think reproducibility is very critical, and that's going to affect very much the fluid flow distribution, and eventually that should have some influence on the cell seeding and the cell attachment. So this is something that is usually taken for granted. So when you do additive manufacturing scaffolds, you expect that they're all the same. But I've shown you that for a commercial scaffold, they're actually not all the same, and very significant difference here. I've also shown you an example on how in silico models can actually enhance cell seeding efficiency by changing the rotation of the scaffold, and I think that really shows the benefit of in silico tools to inform experimental protocols as well. And finally, I think that within tissue engineering in general, I think that there is a need for computational modeling to be part of a new manufacturing practice. so that we actually get much more standardized in terms of the scaffold production. Okay, I'd like to acknowledge the people who worked on this. So Anna Campos was a PhD student at the time and then postdoc in the lab. I also had an excellent undergraduate student, Ralph Goldman, who did the work actually on the rotation. And this was funded by the ERC and EPSRC.
Any questions for Damien?
Yes. I should ask you if you had any plans to validate this system experimentally.
Yeah, I think that's a good point. I mean, we did the experiment, so without the rotation, and I think the results seem to be pretty well. But we haven't, this is new, so we haven't actually done the experiment with the rotation. There is a team in Asia that started to work on doing some rotation experiments, but it's on a different scaffold, so we can't directly validate it. But yeah, the next step would actually be doing the experiment and then see whether that actually works. Yeah.
So I guess it must be difficult to measure the cells by staining and microscopy. I guess that on these scaffolds it must be difficult, but you could measure the flow and look at the impedance of flow and get a measure of seeding that way.
Yeah, that's right. We've also been in the past. What we've done previously is that we've put some osmium into cells, so that we could actually detect them using micro-CT where they were. So we could actually determine very precisely the location of the cells and then count them as well. So that's a way to validate it.
So you did experiment on the texture. You said the scaffold texture and the CAD which you used in the CFD were different texture. Did you see the effect of how roughness of the texture did change the cell attachment?
No, we haven't been. We were not at the resolution of being able to include the roughness. So in all the simulations and in all the characterization, the resolution is too low to be able to actually determine what is the roughness. So we assume it's perfectly smooth.
I may be misreading this, but are we talking about commercially available scaffolds?
Yes.
So what you're saying really is that there isn't a standard quality control type mark.
That's right.
Standard for, so you can bought it from Minipur or you got it from another one.
That's right.
You get different results.
Yeah, and what's in?
We need to control that first before we move to the next step.
Yes, that's what I argue. And what's interesting is that this scaffold, so this is commercially available, but this was actually approved by the lab, the national standards in the States as a scaffold to use, because it's very reproducible, but actually it's not.
If you get a Friday afternoon scaffold,
your
joint's going to be
and I don't want to blame that company because I expect that it's the same for everybody, or if you do it in your lab. So.
More questions?
I can see it.
Hi. We got really nice results from a vacuum seeding method. I wondered if this model would be able to model that sort of seeding technique as well.
Yeah, we could induce some pressure that would suck the cell and then model that in the same way that we did here. So that would be feasible, yes.
More questions? Okay, well, thank you very much, Damien, and good luck with the future.